Hi everyone, today in this video we will be learning about some important questions frequently asked in Neet from genetics, molecular basis of inheritance. So let's begin with our first question. Double helix model of DNA which was given by Watson and Crick was of cDNA, bDNA, ddNA or zDNA. Here the answer is option C, which is bDNA. Also, bDNA is a right-handed DNA having a clockwise twisting, whereas zDNA is a left-handed DNA having a anti-clockwise twisting. Remember that. Now question number 2 says diameter of DNA molecule is 3.4 angstrom, 34 angstrom, 20 angstrom or 90 angstrom. The answer is option C, which is 20 angstrom. 3.4 angstrom is the space between the two nitrogen base in a DNA, and one complete turn of a DNA molecule consists of 10 base pairs, 10 pairs of nitrogen base, okay, which is having a length of 34 angstrom. Okay, these little information sometimes confuses when the question came, so remember it. Now question number 3. Melting temperature Tm value of DNA is high when it contains the amount of pyrimidines is greater than purines, when the amounts of purines and pyrimidines and purines are equal, or when purines are greater than pyrimidines, or when the two are equal. The option is wrong. Okay, here I have already marked the answer, so let's understand the concept. As we know, there is a hydrogen bond between nitrogen bases, 2 hydrogen bond between adenine and thymine, and 3 bond between guanine and cytosine. So melting simply means here how these bonds break, and if there are more number of hydrogen bond, then Tm, which is melting temperature, will be high also. Okay, so the answer must be G plus C is greater than A plus T, which is option C. Okay, and this type of DNA is found in prokaryotes. Okay, and now question number 4 says DNA polymerase 1 is involved in joining of Okazaki fragments, removal of RNA primer, filling of gap, or option D, both B and C. Okay, for joining of Okazaki fragment, there is an enzyme called DNA ligase, so option A is incorrect. Option B and C, which is removal of RNA primer or filling of gap, are the function of DNA polymerase 1. So option D, which is both B and C, is correct. You must learn about the DNA polymerase 1, 2 and 3 since they might become confusing during solving a question, and for NEET purpose this is important. And now question number 5. The enzyme which show polymerizing activity in 5' to 3' direction is DNA polymerase 3, DNA polymerase 2, 1, or option D, all of them. Here the answer is option D, which is all of them. Okay, so remember about these 3 enzymes. Now question number 6. During DNA replication, energy is obtained from. Options are mitochondria in form of ATP, body temperature, from separation of 2 phosphate group from nucleotide, or option D, glucose. Since during DNA replication, the phosphate moiety comes from the nucleotides, which are DATP, DGTP, DCTP and DTTP. Okay, so which are present in the nucleoplasm. So when the bond breaks for the separation of two phosphate group from the nucleotide, there is a liberation of energy which is used during the DNA replication. Okay, so the answer is option C. Okay, it is important for Neet. You must remember this, or it may take time. Now, question number 7. Most stable form of RNA is mRNA, hnRNA, rRNA, or tRNA? The correct answer is option C, which is rRNA. Here, rRNA provides site for attachment of tRNA and mRNA. Question number 8 says, in RNA splicing, non-coding part is removed with the help of Rho factor, SNRPs, RNA polymerase, or none. When you know the terms, it becomes simple and guessing the answer will be incorrect. Okay, so Rho factor helps in termination of transcription. SNRPs stands for small nuclear ribonucleoprotein, which combine with some other protein to form spliceosome complex, which uses ATP to cut the RNA, release the non-coding part, and join the coding part to produce functional RNA. And RNA polymerase is used for transcription. Okay, so the answer will be option B, which is SNRPs. Question number 9. Severo-Ochoa enzyme serves as ligating DNA fragments, protein synthesis, splicing, or polymerizing RNA with defined sequence. Here the correct answer is option D. Okay, and it does in a template independent manner. The polymerization occurs in template independent manner with the help of Severo-Ochoa enzyme. Question number 10 says, Vowly base of genetic code is present at. Options are first nucleotide of codon, second nucleotide of codon, third nucleotide of codon, or any of the above. Here the answer is option C, which is third nucleotide of codon. This happens because normally an anticodon recognizes only one codon, but sometimes an anticodon recognizes more than one codon, and this we call wobbling. Okay, for example, anticodon AAG recognize UUU and UUC, and both these stands for phenylalanine. Okay. And now we have completed with the part 3 of genetics, which is molecular basis of inheritance. Do like, share and subscribe the channel. We'll meet again in another part of the topic. Keep learning.